We can hear you, we can see your screen. So the talk will be about application of the high-dimensional data sets in complex biomedical system at molecular level. Please, the screen is yours. You're welcome.
Thank you very much for the kind introduction, and thank you for inviting me to your 19th International Frei-Konferenz. It is an honor to present before you in these difficult times. I will start giving you an overview of our biomedical data science from physicists for molecular medicine. I am currently researching in the aggressive lymphomas workgroup of Professor Dr. Megiak-Lenz at the University Hospital in Münster. My background is not medicine, though. I'm coming from physics and did my PhD with Professor Dr. Peter Lenz at the University of Marburg in complex systems. And already during my PhD, I went towards complex high-dimensional data sets for molecular medicine and how we can dissect such data sets. And high-dimensional just means that you don't measure just one or a few observables from your samples, you measure thousands of them. And as promised in the program, later on I will talk a bit about how to quantify and dissect such complex biomedical systems at the molecular level. But let me start by giving you a motivation of why we are studying cancer cells, as you can see here, stained in blue under a microscope. You might know that cancer is a genetic disease caused by mutations in the DNA of healthy cells, causing them to lose their normal function, and they have no natural cell death anymore. And because they are still very similar to healthy cells, they can evade the immune system very often. And this gives them the power to grow uncontrolled in the human body, which is the biological basis why cancer is a leading cause of death. Already in 2000, there were over 10 million new cancer cases diagnosed globally. 2018, it was 18.1 million. And in part due to an aging population, it's predicted that from 2040 on, there will be over 27 million new cases per year. And if these patients are not treated, almost all of them would die. So all of these patients need medical help. And the bad news is that even if they are treated, many will still not survive. Millions will die per year, which is in part due to a chemotherapy that can be toxic because it's working like a broadsword that also kills many healthy cells, and in part due to emergence of resistance in the tumor cells during therapy. So most of these patients will need better help than medicine can provide today. And our overarching goal is to go from broadsword to scalpel to precision medicine, which means that for each individual patient, we want to find specific attack vectors to just kill their individual cancer cells and not healthy cells. And what we've just seen in the last talk is also encompassing in this big goal, which is precision delivery of drugs via nanoparticles. But I will today talk about how to understand the cancer cells and their inner workings better. So after this motivation and our goal to understand cancer at the molecular level, let me show you what we can actually measure. So typically in hospitals, medical practitioners take 3D tumor biopsies from a patient. And this is a 3D bulk of many different cell types. So one big challenge in this context is the extreme heterogeneity of the samples. There are cancers, even if diagnosed with the same medical name, same diagnosis, are different between patients. And there are many different cell types within the tumor probe of the same patient. To give you an example of how complex it can become, have a look at this interaction graph. You don't have to understand all of it. Honestly, I also don't understand all of it. Just give you an example. This is a cancer cell. This is a T cell. It's part of the immune system, T regulator cells, macrophages, natural killer cells, and all these different cell types are part of the tumor sample. It's not just pure cancer cells. And for example, this TCR stands for T cell receptor. It's a macromolecule on the surface of the T cell, and it can bind to another macromolecule that may be on the surface of the cancer cell. And this is a mechanism how cancer cells could theoretically evade the immune system, so not being attacked by the T cell. But just keep in mind, it's a complex interacting system, but we need this level of understanding ultimately in order to reach our goal for precision medicine, like triggering cancer cell self-destruction or making the cancer cell visible as a target for the immune system. So now that you have seen the samples we can measure and how complex it can be, let me start by giving you an overview of our biomedical data science on the genetic level, DNA level. So let me briefly recapitulate what is DNA. You might know that DNA encodes our genome. It's located in the cell nucleus. It's a quite astonishingly robust macromolecule made of so-called base pairs. For example, you see here the chemical formula of the guanine base, and here the formula of the cytosine base that are hybridized together, not covalently bound, but still strong bounding. And there are such strands, long molecules, which are not completely shown here. And you can have such base pairs, many base pairs in a row. And to give you an impression of how large and how many information the DNA has, the human reference genome overall comprises 3.2 billion base pairs. And to better imagine how long this is, imagine a string of letters A, C, G, T for these 4 bases. If you would want to have the complete reference genome in the computer, it would be a 3 gigabytes long string. So it's quite a lot of information. We focus on the exome. This exome contains the protein coding genes, about 20,000 in humans. It's only 1.2% of the genome, but it's the most important 2% because these proteins, which these genes code, are the macromolecules that confer the function within the cells. And now that we know the target of measurement, how do we measure it? Fortunately, this is already industrialized, so-called next generation sequencing. Here you can see a commercial measurement device, Illumina NextSeq 1000. It costs about 200,000 euros. And you have protocols, for example, for sample cell preparation. The DNA, this long molecule, is randomly split into short pieces. When these molecules are amplified, you get many copies of them. This prepared sample is inserted into the machine, and then it measures molecules of up to 100 base pairs in length. And our work really starts then with the raw data from these machines, which are called reads. And in the computer, these are just strings again with letters A, C, G, T for these bases of length 150. But we get millions of these reads per patient. The next big step then, once we have these sequenced DNA reads for many patients, is that they enter our analysis pipeline for primary analysis. This is taken from our publication from 2021, where we studied plasmaplastic lymphoma. And the first step is alignment, because we do not know from these 150 base pair short reads to which genes they belong. So we need this 31 gigabyte long string as a kind of reference database and a method like HESAT to actually find out where each of the million reads matches to the reference genome, to which gene they belong. Once this very computationally heavy computation step is done, we mix it and add additional metadata, for example, information, does the read come from a tumor sample of the patient or a normal baseline sample? This is the best way of studying these because you have many variants in the normal baseline. You have to know we all have different sizes of ears, different eye colors. We have many differences. So every one of us has millions of differences relative to the human reference genome. And of course, we are not interested in these differences because they are not specific to the cancer cells. So we have to somehow filter them out. And the first part is to use big databases of germline, and germline means known variants in the human population relative to the reference genome. And this goes into variant discovery. We use for this the genome analysis toolkit from the Broad Institute in Boston, namely their MUTACT algorithm. And it gives us, in the simplest case, single nucleotide variants, which means just one base is replaced by another base at some position in the genome, or insertions or deletions, which means that multiple consecutive bases, for example, have been removed relative to the reference genome. And for this plasmablastic lymphoma study with about 100 patients, this initial step gave us 17.1 million raw variants relative to the reference genome. And as I already said, we are interested only in the cancer-specific variants, which requires us to use a sophisticated filtering pipeline to get to them. There are some basic filters that remove artifacts or things that are not statistically significant, but then you still have 40% of the 17.1 million available. Then you can use other big databases of normal variants and variants you do not want to filter out because they are known to be pathogenic in cancer, from the NCBI in the US, for example. You annotate all these variants, perform advanced filtering with these databases, and then you are already down to 0.4%. It's still too much, but most of them have been filtered out by now. And then you do the computationally heavy functional annotation. So far we were on the DNA level, but biologically the important thing is what happens or what is the effect of a DNA mutation on the protein level, because the proteins are the functional molecules in the cell. And another key method we use together with gene models from RefSeq and another database of principal transcripts is transvar, which gives us the impact on protein level. And with this impact, and also requiring that at least 10% of the cells in our tumor sample carry a certain mutation, we then end up with only 0.09% of the variants relative to the reference genome that we really call somatic mutations. That somatic means mutations gained in the cancer cell relative to the healthy cells you have since birth. This was an example of our analysis pipeline for the genetic level. Now let's have a look at the results, in this case from our Burkitt lymphoma study, which we published last year. And every column is one patient, every row is one gene, and in this Burkitt lymphoma study, the ID3 gene was mutated most frequently with 76% of the patients. The colors are different types of mutation, not important right now. And this primary analysis results in form of the somatic mutations then is typically used in higher level statistical analysis. For example, these are just other studies for follicular lymphoma, which we published last year, and PBL, which is the paper if you want to look for details for the analysis pipeline from the last slide, you can find it in this paper. And such higher level statistic analysis, for example, take these mutations and look over the age spectrum of the patients. Here you have the children, 11 years, and here you have the elderly. And for example, this ID3 gene, we found that it is predominantly mutated in children, not in the elderly, whereas the elderly have other genes that are mutated, which are almost not mutated at all in children, which is just another example of heterogeneity, although all these patients got medically the same Burkitt lymphoma diagnosis. But such heterogeneity gives possibilities for further study and towards precision medicine, towards scalpel, towards better treatment. This was the genetic level. Now let me give you an overview of the gene expression level. So we go one level up now, not just the DNA. You have to know in the cell, the DNA is transcribed into so-called messenger RNA molecules according to the cell's specific programs. And these messenger RNA molecules are then later translated to build the proteins, which are the function-carrying molecules in the cell. So briefly, you can just remember mRNA is the actually used part of the DNA. And that's information, that's very rich information biologically, because most of the genes in the DNA are not used by a certain cell type, either healthy cell or cancer cell. The measurement of gene expression and the primary analysis are fortunately nowadays more or less the same as I already showed you for the DNA level. It's even the same measurement device. And also the alignment step to find out where the reads, to which genes the reads belong, is just as for DNA. But then we are not interested in variants or mutations relative to the reference genome. Instead, we just simply count the reads for every gene position or for every transcript, which is a bit more detailed than genes, and humans have about 55,000 known transcripts, and every row in this resulting matrix of relative intensities is then one transcript. And in this case, in this example, it's for 500 tumor samples. And you have to zoom in quite a bit, wait a bit, until you actually can see individual measurement values. And the message here is that it's large, like a 27 megapixels photo large, but it doesn't have any neighborhood as in a normal photo, like large green forests. And it contains a lot of random and systematic noise. It's a very complex signal, also due to the true heterogeneity that underlies it. Just remember, per sample, we measure an ensemble of many different cells at the same time. And even within each cell, you have many concurrent gene expression programs, and you measure their net effect if you measure the sample. And in this, or from this large complex signal, we want now to discover useful signatures of biological interaction where we can learn something from. Let me give you an example of such a useful signature. You see again the cancer cells. They are from diffuse large B-cell lymphoma, and unfortunately, alone in Germany, there are 5,000 new cases each year, and if untreated, the disease ends deadly. And already in 2000, Alizadeh and others discovered by hierarchical clustering a pattern between different tumor samples of this disease, and by comparing these patterns with healthy activated B cells or germinal center B cells, they saw a similarity. And once you have found such a signature, which corresponds to a specific subset and ordering of genes, you can sort new patients, every column is a patient, by the signature, as done here by Visco and others in 2011, from ABC-like DLBCL and GCB-like DLBCL. And over this sort order, which you cannot define if you just have the microscope image you just saw before. You can only define it if you really look into the cells via molecular measurement. Is clinically important because if you look after 5 years, you will see that with the exactly same therapy, all these patients got the same therapy, still 70% of the GCB-like DLBCL patients live, whereas only 40% of the ABC DLBCL patients have survived. So we obviously can learn from such molecular heterogeneities, and ultimately the idea and goal again is to use these heterogeneities to understand them sufficiently to define specific therapies for these different subtypes of patients in the same disease. But recall, we first have to discover such signatures in a huge, noisy, high-dimensional matrix. Let me now give you a theoretical, idealistic view on signal complexity. We start with a matrix where the columns are samples, where the rows are genes, and initially it's a zero matrix, zero signal. Now let's simulate a signature that resembles the true signature you just saw for some subset of samples and some subset of genes. And now you have to see that we initially do not know which subset of samples or subset of genes is affected by some biology. So in the simulation, we have to destroy this information by randomly permuting the gene rows. This results in this. And also randomly permuting any two columns. And the same signal looks like this. And now assume we have overlapping, that many concurrent biological effects are in the same cells, with overlapping signal. Let's simulate the same signal, just additive in this new order, in this new permutation of genes and samples. And again, destroy the information by permuting gene rows and permuting sample columns. And we do this several times. And then we add measurement noise, also simulated. And if I had shown you this signal, the simulated signal, initially, you would probably have said, nah, I cannot really discover any structures in there. But if you knew where to look, at which permutation and order of genes and subset of an order of samples to look, then you could still find all the 7 simulated signatures just by reordering in this high-dimensional signal. And this is just a simulation of 0.2% of the true gene expression matrix we measure. In order to find them, that's the difficult part, we need a search strategy. And we do not only want to find them, we want to extract their true noise-free contributions. And to do this, we will need a signature model and regression model, which I will show you soon. Now I come, as promised in the program, to dissection of high-dimensional datasets, quantifying complex biomedical systems at the molecular level. More precisely, I come to our method signal dissection by correlation maximization, SDCM. If you want to know all details, you can have a look at our paper from 2019 that's based on my PhD. And if you want to apply SDCM to your own data, you can find the source code at github/GrauLab/SDCM. But today I'll try to give you a high-level conceptual short overview of how the method works. Let's start with the theory. So this matrix you just saw is simply modeled as a matrix R to the power of m genes cross n sample columns. And we give names to the initial, not very meaningful measurement order of genes, I0 from 1 to m, and the sample, the column order, J0 from 1 to n. And the signal model is relatively straightforward. We want to model this matrix as a sum k from 1 to k hat of ek of matrices, signature matrices of the same size as m, plus some noise term. And our task, or SDCM's task, is to reverse that sum operator. So we only have m, and we want to discover the summons. And this, of course, is a mathematically highly ambiguous problem, and we need additional constraints. Constraints that are mathematically sufficient to enable this reverse dissection, but they should also be as flexible as possible to model and discover as broad as possible natural phenomena that might exist in the data. This brings me to my signature model. I demand bimonotonic consistency. Let me briefly explain what this is. So for any signature k, I demand that there exists a permutation, a reordering of the genes and a permutation, a reordering of the samples, such that in this reordering, every gene row of the EK matrix is monotonic, either monotonically increasing or decreasing. And also every column is monotonically increasing or decreasing in this reordering. And due to this consistency demands, you can see that any 2 rows or any 2 columns are automatically correlated or anti-correlated, which is what we are looking for biologically. And if I have to graphically summarize both the signature and the signal model, I would depict it like this. You start with this noisy matrix and want to model it as a sum of overlapping bimonotonic signatures for yet to discover, and that's the difficult part, yet to discover suborderings, reorderings, permutations, subsets of genes and samples. And you might know that the number of subsets of genes, of n genes, grows like 2 to the power of n. So we really need a search strategy. We cannot simply test all possible subsets. And let me now show you concepts in a just 3-dimensional example to better visualize it with just genes x, y, z, how SDCM tries to discover these effects. And you have to remember SDCM only sees the coordinates of the points. The colors are just for you to better understand the simulated effects. And there are 4 steps. The first step is that an initial representative that corresponds to a certain direction in the gene space is discovered. Then additional representatives are used to optimize that direction until it's locally converged. Then bimonotonic regression, the consistency model, is used to regress this bimonotonic curve. And once it's discovered, it's dissected, it's subtracted towards 0. Now let's go through the same 4 steps a bit slower, a bit with more detail again. The first step is to find such an initial representative, which is done by a signature functional. For any direction in gene space, it gives you a number, and this number is higher if more sample points are along this direction, and the more correlated and more near to the line these sample points are to this direction A. And once found, we use a correlation matrix, X in A over norm X and norm A, which is a number between -1 and 1. You might have heard cosine similarity, that's the same. And I need a bit more flexibility because of weights, which correspond to the signature focus, which I will show you now, to optimize this direction by taking additional representatives and defining which points have a high weight, which are nearby this direction. And geometrically, the signature focus, these weights are just this double cone. All points inside the double cone have a high weight. Now, let us, once you have this direction in the gene space, you can project all of the data points onto this direction, which gives you an ordering of all sample points. And in this ordering, now let's change to the heat map view. In 3D, it's easy. In high dimensions, it's necessary because you no longer have a 3D view. You only have the heat map then. And here you see again the genes X, Y, Z. Every column is 1 point in the upper view, and the order of columns is by this projection order on the line. For example, these green points have high Z coordinate and low XY coordinate, so these columns correspond to these green points. And in this signature order, now we use again the bimonotonic consistency model, which transforms every line and every column in a monotonically increasing or decreasing function. And the columns that are gray are just the points outside of this double cone, outside of the signature focus. They are not used for this regression. And the signature curve is now smooth. You can draw it again in the 3D view. It runs through this point cloud. And now the next step again is dissection of the now explained signal, which we can subtract towards 0. And trying to leave the other signal part still in the signal for later iterations. And the next iteration discovers then in the same steps this red effect. And the next iteration discovers this offset effect and subtracts it. And then for step 1, it is detected that there are no significant signatures left in the signal. And SDCM terminates and gives you the results. Here you see the results for this 3D concept example again, these 4 curves, 4 effects. And in the paper, of course, we compared this with many existing methods like principal component analysis, independent component analysis, non-negative matrix factorization, hierarchical clustering, bi-clustering methods like Fabia. And from all these methods, the good old principal component analysis, which you might as physicists also know as the main axis of rotation from mechanics, was the second best for the high dimensional data sets. So let's compare SDCM and PCA conceptually. Here you see for the same data points, the 3 principal components, and the first principal component points in the empty space between the blue, red, and green effect, because that's the direction of maximum variance, which is what principal components look for. SDCM, in contrast, looks for direction of maximal signature function or maximal correlation. Whereas SDCM discovers monotonic nonlinear curves, PCs are always lines. And because of the reduction step that principal components use, which is full projection mathematically, all the principal components are orthogonal to each other. Whereas SDCM only dissects the explained signal in the double cone, in the signature focus, and then is also able to discover 4 effects in a 3-dimensional subspace, whereas there only can exist 3 principal components in any 3-dimensional subspace. So far for concepts and for the simple 3D example. Now let's go high dimension. As I showed you in the simulation earlier, I have now simulated several signatures, this time of different size and form. The first signature comprises all genes. This is an overlap signature. This signature has very low signal at the edges, at the corners. And all these signatures are permuted for random order suborderings of genes and samples and added with noise to form this input matrix. And SDCM now has the task from this high-dimensional matrix to rediscover the simulated signatures. And as this is our test case, of course, it was able to do this. So you see it was able to discover all 7 signatures. This is a bit rough because at the corners, it just has a signal strength of the level of the simulated noise level, but it's still discovered. And to measure the quality, we use such correlation matrices where every row corresponds to one of the simulated directions in gene space and every column corresponds to one of the discovered directions in gene space. And red means correlation almost perfect, near 1. Black means no correlation, almost orthogonal. And we do not want such gray things because it means that one simulated signature is explained partly by more than one detected signature. And we want to have one-to-one relationship. SDCM does this in this case almost perfectly. PCA also discovers the first signature very well and very strong with black off diagonals. But it mixes the signature 2 and 4 in this example and signatures 5 and 6. So the sensitivity and specificity of SDCM is higher than principal components analysis already for this relatively simple 7 signature example. Now let's turn up the difficulty. Now I simulate of pattern 3, 16 signatures in, and again give this signal into either SDCM or PCA. SDCM is a bit weaker now with a bit more gray pixels, but rediscovers still all 16 signatures, whereas the principal component performance broke down essentially, which shows that SDCM can help you discovering and extracting in a one-to-one relationship specific signatures from so high-dimensional signals. You might know clustering or might even have used clustering already, like hierarchical clustering. The same signal input into hierarchical clustering gives you this plot where you can see 2 clear clusters in the samples, 1/3, 2/3, and 2 clear clusters for the gene rows, 1/3, 2/3. And this is caused by the large signal of signature 1, which I showed you in the last slide. So signature 1 dominates the hierarchical clustering. And that's a weakness many cluster methods have. They have no signal model. They have no ability to really modify the measured values and model them as a sum of something and then test again. They have no dissection step. They only give you one global ordering. And if it's dominated by one signature, you have just the signature and nothing more. And UMaps you might know as a useful tool to project high-dimensional data and visualize them in 2D space. To keep the topology, to keep the distance between your objects. But if you do this with the 1,000 gene rows of this signal, you again end up having 2 clusters, which is clear. One cluster corresponds to the lower half of genes, one cluster corresponds to the upper half of genes, and it doesn't have the ability to discover differences across the clusters or shift the clusters over each other. So that is why we need dissection. We want to peel off such overlapping signals, which could also be a technical measurement artifact overlapping the true biological signals, for example, and thereby merge the initially split similarities across already detected clusters. And the idea is, as shown on the last slide, to get then a higher specificity of discovered signatures with the real causal sources in your signal, which is key for interpretability, not just in biology, but probably in any case with such high-dimensional measurement values. So far about a conceptual overview of how SDCM works. Now I come to the last part of my talk where I will show you some signatures discovered by SDCM for real gene expression data of diffuse large B-cell lymphoma. One signature which I discovered always in transcriptomic datasets looks like this, a very strong binary signature. And if you combine it with metadata about gender, these are the women, these are the men, you can identify it as the gender signature. You can also do a bit statistic and ask how probable it is to find such a signature just because of random data, and it's almost zero, so it's validated. And of course, the gender information is not part of the input information to SDCM. SDCM only gets the gene expression values. It's a good control signature. SDCM also rediscovered the cell of origin signature from the introduction with the GCP-like DLBCLs on the left and the ABC-like DLBCLs on the right. Again, it cannot be random that it is discovered. And let's revisit again the monotonicity argument or bimonotonicity consistency model. If we slice this matrix here and look at one of the top genes, and these are now the pixel values, you see that it's quite noisy. So the order of samples is not sorted just by this one top gene. But if you look for other top genes, you always see the same monotonic trend. So the signatures discovered by SDCM really work on gene sets level, which is much more robust than if you would work on a single gene level. And of course, there has to be some kind of distance between gender and this signature that in the data that is used by SDCM indirectly. And to visualize this, now let's draw this gene over the gender order over this x-axis, which is the sample order from GCP to ABC-DLBCL. And then you can see the values of these pixels are no longer monotonic at all. They are completely uncorrelated. And this is the distance in the data we make use of to dissect signatures. SDCM also detected previously discovered stromal 1 and stromal 2 signatures from 2008, but we are most interested in signatures that are able to predict patient survival under current therapy. So next in the paper, I took all signatures discovered by the SDCM, all principal components, and all the signatures discovered by other methods, looked at any two of them, any pair of them, and put them into a so-called proportional hazard model, which fits patient survival. And I asked which of the pair of signatures can predict patient survival best. And the answer was this pair of two SDCM signatures. It could also have been one SDCM signature and one independent component, but the second best model only had a relatively likelihood of 0.02, which again underlines the specificity that SDCM can deliver. And let's have a look at these two survival predicting signatures again. First, we validated them now on gene expression level in two cohorts, one measured with microarrays, one measured by RNA sequencing as shown in the introduction. And you can see for the same ordering of genes, you can order the samples in a similar way in both these independent DLBCL cohorts. So it's definitely validated to exist also in independent cohorts of diffuse large B-cell lymphoma. And all these three cohorts had measured the outcome, the survival of patients. And if we associate with this, you might not know these Kaplan-Meier plots. You have a time axis on the x-axis and a survival from 0 to 100%. At time of diagnosis, all people per definition still live. And as you go over time, people die and the curve goes down. And with help of these signatures, we can group the patients or order the patients. And if we look at the 4 quartiles in this ordering, then you see a trend from low survival to high survival. And you see the same trend independently in all these 3 cohorts. So it's validated to be really a clinically relevant effect also in 2 other independent patient cohorts. Especially if you compare this with the previously known GCP and ABC-DLBCL curves, you see that you can now predict survival differences much more stronger. And the interesting part was if we now went into the subtype of GCP-DLBCL, about half of the patients, and again applied our predictor, then we were able to even show in all 3 cohorts a much stronger difference in survival. And 1/4 of the subtype, which was the 70% subtype from the introduction, so the one that is thought to be of better survival, actually has 1/4, 25% of patients that have lower survival. And again, heterogeneity, again, these patients would profit from a more targeted, more specific therapy. And that's the basic work for that. And by now, this 25% is also recognized as new clinical subtype in diffuse large B-cell lymphoma. And it's just the patients in this heat map view that are on the right side of both these heat maps in any cohort. This brings me to my summary. I've shown you first on genomic level, DNA level, that we can, via a long analysis pipeline, discover somatic mutations. With these somatic mutations, we can then associate, for example, patient age and find heterogeneity within, in this case, Burkitt lymphoma between the patients. And on the gene expression level, the amount of DNA that's actually used by the cells to build proteins, I've shown you SDCM as a tool to discover signatures that are discovered with a general mathematical model. So it doesn't necessarily have to be biological data that you put into SDCM. And signatures in the DLBCL context that were able to predict strong differences in patient survival. And all these analyses together, I would call modern analysis for molecular medicine, short MAM. And some of you might know that we are currently applying for a German-Ukrainian collaboration in this MAM context. And I would like to take the opportunity to briefly thank the physics side in Ukrainian, the physics side in Germany, the medical side in Ivano-Frankivsk, and the German side in medicine. Too many to name them all in detail. Just let me thank Georg and Lubomir as coordinators and representatives of the respective side. And now I want to thank you, the audience, Yakuyu, for your attention. And I'm looking forward to your questions. And of course, I'm also happy to connect later, for example, via Twitter, now X, at Dr. Michael Grau, or if you want to use the source code and dissect your own data via GitHub/GrauLab/SDCM. Thank you very much. Yakuyu.
Thank you, Michael. I guess we have time for questions. Please, do we have any questions? It was very interesting talk about very smart algorithms, about big data and so on. So please, if you have any questions, maybe from online audience, we have more than 20 people there. Please raise hand or just ask to you. I have just a technical one, maybe. What kind of computer power do you need to perform such algorithm routinely? I mean, do you really need very powerful computers, or this can be done on some kind of more or less whatever, simple clusters or something like that? Do you really need very powerful things?
It really depends on the input size of the matrix. So the smaller samples, examples like the 3D example, and also the 100 by 1000 input matrix in high dimensions runs on your average desktop computer, no problem. They are also the test cases which you can, if you download the source code, you can directly execute on your local workstation. But if you go to larger matrices like 50,000 transcript across 10,000 patients, what we try now in the pan-cancer situation, then you quickly come to memory limits. Memory limits are actually more important than CPU limits in this case. It's still possible, but you really have to use a workstation or a cluster node with 500 gigabytes of memory then, and it also takes several weeks to fully compute the signal. But this depends a bit on how complex the signal is. I mean, if it was a pure noise matrix, then it's much faster finished because it doesn't detect any signatures. But the more signatures are overlapping, the longer it still discovers something that's significantly not noise. And the more signatures are discovered, the longer it runs before it terminates.
Okay, great. Thank you. Any other questions, comments? Don't see any so far, so thanks a lot, Dr. Grau, again.
Thank you too. Bye.
Yeah, so we have one more talk, which will be ordinary talk, and it's from Professor